Hello. So last time I left off, I finished up the applied immunology stuff. Talked about monoclonal antibodies, Western blots, and phage display libraries. And this content's maybe a little easier than that. Antigen presentation. So we're going to look at some of these things associated with antigen processing and presentation. So just a little bit of background on these molecules involved in this. The T cell receptor is related to the B cell receptor evolutionarily, and it's most likely that the B cell receptor evolved from the T cell receptor. Both of them have V and C regions, which are variable regions and constant regions. And rearrangements take place in a similar sequence of events to the B cell receptor for the T cell receptor. And we'll talk more about that when we talk about T cell development and B cell development. But the key difference is that the T cell receptor, at least in the case of alpha beta T cells, which are your normal T cells, the T cell receptor is built and selected for so that it can bind to MHC molecules, MHC1 or MHC2. Now, MHC molecules, we've talked about them, a good bit, and you at least got a little bit of a structural picture of what they look like. But maybe it's due time that we talk about what they actually are and where they originated and all that stuff. So MHC proteins, MHC molecules, they are transmembrane glycoproteins that are encoded in a large cluster of genes called the major histocompatibility complex, MHC, that is located on chromosome 6. Now this MHC complex of genes was first identified in genome-wide association studies when they were looking into what factors play a role in the rejection of tissue transplants. So they found, if they could match somebody's alleles for the MHC genes to a donor, then that donor would not be rejected by the recipient. So that's where they were originally characterized. They didn't know what they did at first, though, of course, and that has come a long way since then. So the peptide binding clefts of the MHC molecules are extremely polymorphic within the human population. So there are like thousands and thousands of different variants of each MHC gene, MHC1 and MHC2, and there's 3 of each of those. So most people are heterozygous for the alleles at each MHC locus for each MHC molecule. So they actually have 12 total unique MHC molecules on their cells. Now remember that an allele is a slightly different version of an individual gene. It's still the same gene, it's just got a couple of mutations that are different. T cell receptors, they both bind to the peptide that's been loaded into an MHC protein, and they bind to a little piece of the inside of the MHC molecule itself. And so this introduces another dimension to antigen recognition by T cells that is referred to as MHC restriction. So a single T cell receptor is specific for a specific peptide, a particular specific peptide, bound into a specific type of MHC molecule. And that molecule is going to be encoded by a specific allele variant of a specific MHC gene. Okay. So since most people are heterozygous at each MHC gene locus, that means that every MHC molecule type will have 2 corresponding subpopulations of T cells, each with specificity for that MHC protein that is encoded by the allele on that chromosome. So basically, you inherit a chromosome from mom, and you inherit a chromosome 6 from dad, right? And so for every MHC1 protein you have from mom, you're going to have a different one from dad. So for every MHC1 gene you have, you actually really have 2 copies of it, 2 unique copies of it, because almost everybody is a heterozygote at every locus. Okay, and therefore you have 2 different sets of T cell receptors that are each of which is specific for either mom or dad. Okay, so you basically have mom-specific and dad-specific T cells that recognize the proteins encoded by the alleles that you inherited on chromosome 6 that came from your mom and dad, respectively. Evolutionarily speaking, it's likely that MHC evolved first, and it probably evolved as early as the sponges first evolved, which are the simplest animals. And then the T cell receptor evolved, and it was able to, recognize MHC and distinguish foreign from self. And then immunoglobulins finally evolved, and they evolved, I believe, around the time that birds evolved out of the animal lineage. Every one of these proteins is a member of the immunoglobulin superfamily, which we learned about for adhesion, but it also applies here. Okay, so a little bit about T cells and such, and more elaboration on this stuff. Alpha beta T cells, they recognize antigens as peptide MHC complexes, and you already know that. Those peptides, you may not have thought too much about this, but those peptides that are loaded on the MHC molecules, they can come from self-proteins, they could come from intracellular pathogens, or they could come from pathogens that have been taken up from the extracellular space, right? However, T cell receptors don't usually recognize self-proteins, and there is a complicated reason for that. They are selected against during development. If they are self-reactive, they are put into apoptosis. Okay, so you don't really have self-reactive T cells. They usually only respond to foreign peptides in MHC. Otherwise, you would have an autoimmune issue where you're targeting your own proteins. So, in somatic cells, in most tissue cells, peptide MHC complexes will signal the presence of an intracellular pathogen that needs elimination by armed effector T cells. Right? So MHC1 is expressed on all tissue cells. The only cell that doesn't express MHC1 is red blood cells. And that means that any pathogen that is invading a cell in your tissues can be targeted by a cytotoxic T cell, right? Specific to that pathogen's antigen or that pathogen's peptides. In dendritic cells, peptide MHC complexes don't really always signal the presence of an intracellular pathogen, right? What their job is to activate antigen-specific naive T cells. So they go to the lymph nodes and they activate naive T cells and they say, hey, there's a problem, here's co-stimulation, here's antigen, and you should expand and turn into an effector cell that can take care of this problem. Together with co-stimulation, this delivers activating signals to the naive T cell, and that induces differentiation into effector T cells, right? This is all probably review. Other classes of T cells are sort of interesting. So I've been talking about alpha beta T cells this whole time, and those are your MHC restricted T cells. But there's also other weird types of T cells that we'll talk about more in the T cell chapter, but we'll talk a little bit about them in this PowerPoint. We've got these MIAT, the mate T cells. We have the gamma delta T cells. These don't recognize MHC molecules, technically speaking. They don't recognize the classic MHC1, MHC2. Instead, they recognize different types of surface molecules, and often those surface molecules are expressed by cells that are infected or under extreme amounts of stress, either due to infection or because they are cancerous or because they're just, in a bad condition of some kind. So, one example of a ligand for the mate cells and the gamma delta T cells are these class of proteins, these group of proteins called the non-classical MHC class 1 proteins. So we've talked about MHC in this class so far, but we're referring to the classical MHC1. There's another set of genes that encode weird MHC1 proteins that really don't do the same thing that MHC1 does, and they are called non-classical MHC class 1 proteins. We're going to talk about a good number of those, but only a few are going to be important. Some of these non-classical MHC1 proteins act as ligands for mate cells or gamma delta T cells. They might act as ligands for the receptor NKG2D that is expressed on T cells and natural killer cells. That can target them for destruction because this is a receptor for, danger, essentially. It is a receptor for stress proteins. And then there's also this class of cells called the invariant natural killer T cells, the INKTs. These are cells that recognize lipids, presented by CD1, which is one of the MHC class 1b proteins, or the non-classical MHC1 protein. Okay, so we're going to elaborate on all of these in due time. Well, let's look first at the classical MHC1 and MHC2. Here's a really pretty figure. There's only really so much you can learn about MHC1 and 2. So MHC1 is a one-legged molecule, and what I mean by that is it only has one polypeptide chain. Okay, so it's literally only one-legged, and it only has one transmembrane domain. So it's one-legged, so it's not very stable on the cell surface. Even when it's loaded with a peptide, it's still not very stable on the surface. So what it has to do is during the production of MHC1, it has to associate with this little nifty protein called beta-2 microglobulin, which is basically a protein that serves as a crutch to keep MHC1 upright on the cell surface and folded properly. Now, the real job of beta-2 microglobulin is to just help proteins fold, and MHC1 happens to be one of those proteins. But this is the way that I always remembered it, is MHC1 is one-legged, so it's not very stable. It would fall over, but it needs this crutch, this beta-2 microglobulin, in order to stay upright. CD1, the polymeric immunoglobulin receptor, and several other proteins also bind to beta-2 microglobulin and require that action in order to show up on the cell surface. So this MHC1 is not the only protein that binds to beta-2 microglobulin. There's other proteins that do it as well. Some of which are immunological, some are not. But here you can see the peptide binding cleft. This is like a view from above the MHC1 protein. So the peptide would sort of sit in here like a hot dog. That's what Sanders always says, at least. So MHC1 peptide complexes are antigens recognized by cytotoxic T cells, CD8 positive T cells, right? You've seen these figures before. Here you have a cytotoxic T cell, and its T cell receptor is binding to a peptide antigen loaded into MHC1. CD8 binds to the outside of MHC1 to stabilize that connection. And this is how antigen recognition occurs in cytotoxic T cells. And then this figure shows you how the MHC1 binds to the CD8. Okay. On the other hand, MHC2 is a two-legged molecule, and it is stable on the cell surface, so it doesn't bind to beta-2 microglobulin. It doesn't need to. And it has a similar peptide binding cleft. I believe it actually binds to peptides that are a little bit longer than the MHC1 peptides. It has a predilection to bind to longer peptides. And of course, MHC2 peptide complexes are recognized by by T helper cells, CD4 positive T cells, right? And CD4 binds to the outside of MHC2 during antigen presentation. That should be reviewed. Okay. So MHC2 peptide complexes, those are the antigens recognized by CD4 positive T helper cells. And this is a figure showing you the identical thing as the slide on cytotoxic T cells. And then this is what it looks like in a little bit more of a structurally accurate depiction. Still a cartoon, but this one is more of what it looks like. So it's sort of showing you the same thing here. So before we get into how these are all made, these MHC proteins are made, we should probably know some of the more important features of them and some facts about them. We know basic stuff, but we got to know a little bit more. So first of all, MHC molecules that lack a bound peptide are very unstable and they do not fold properly. They do not make it to the cell surface, and if you put them on the cell surface, they're going to collapse inevitably and they will be taken up by the cell and degraded. Okay, so they need to bind to a peptide to show up on the cell surface and to remain there. MHC1 molecules binds to peptides that are 8 to 10 amino acids long. Now, with that being said, longer peptides can initially bind to the MHC1 peptide binding cleft, and that is up to 16 amino acids those peptides can bind, but they're going to be trimmed down to the 8 to 10 size after they bind. Okay, so the MHC1 binds to short little peptides, 8 to 10 amino acids. On the other hand, peptides that bind to MHC2 molecules, they are usually 13 to 25 amino acids long. Okay, so the one with the higher number, MHC2, binds to longer peptides, higher number of amino acids. MHC2 can also accommodate a greater variety of basically peptides than MHC1. MHC1 is fairly restricted in what can bind to it. MHC2 is really not restricted in what can bind to it. And we're going to look at what restricts these peptides and whether they can bind or not. Remember, all nucleated cells and platelets express MHC1, and only certain cells express MHC2. So that's going to be your myeloid phagocyte system, your macrophages, monocytes, and dendritic cells, but also your B cells. And under the influence of interleukin-1, ILC3s and ILC2s can also express MHC2, as well as thymic cortical epithelial cells. We'll learn more about what those do later during T cell development. And also activated T cells, but not naive T cells. Weirdly enough, they can express MHC2. And many other cells only under the influence of interferon gamma. For example, fibroblasts, endothelial cells, epithelial cells, all of these things can express MHC2, but only when they've been stimulated with interferon gamma. Remember, interferon gamma is one of those cytokines made by T helper type 1 cells, and it's made mostly during viral infection. MHC molecules of both types can be recycled from the cell surface, and they can be reloaded with a new peptide. However, they need to last a good long while before this happens, right? They're going to stay at least several days, probably a week or two, on the cell surface, so long as their peptide remains bound. If the peptide falls out of the MHC binding cleft, then yeah, it's going to be recycled. The MHC molecule is going to be taken into the cell and reloaded with a fresh peptide. On the other hand, you don't want to do that because hopefully dendritic cells taking up antigens in the tissues can carry them all the way to the lymph nodes, and that can sometimes take a good week, maybe even a little longer, depending on the situation. Right? So those MHC peptide complexes, they have to last a very long time on the cell surface. So they have to be binding to each other very tightly in order to not fall out, in order for the peptide to not fall out of the MHC cleft. Okay. Some more important features. Most of these might be reviewed for you. MHC1 is important because it binds to the killing inhibitory receptor, the KIR class of receptors on natural killer cells. And that tells the natural killer cell to not kill that cell. Okay? Natural killer cells will kill any target cell that has an abnormally high ratio of activating proteins to inhibitory proteins on the cell surface. Okay, so pretty much all cells express some level of activating and inhibitory proteins. But if suddenly cells become very stressed out, and or if they suddenly stop expressing MHC1, or they express it a lot less, natural killer cells raise an eyebrow, and they come and they kill that cell. They recognize that something is wrong with it. Better to just kill it than take the risk. Okay, that can shut down viral infection, and that can also kill cancer cells before they have a chance to get established. MHC1 is one of the most important inhibitory signals for natural killer cell activity, but another one is HLA-E, which is one of those MHC class 1b molecules, those non-classical MHC1 molecules. And we'll learn more about HLA-E later on. And some more important features. MHC1 molecules, they are loaded with peptides in the ER of the cell, the endoplasmic reticulum. Okay, so peptides derived from the cytosol are actively transported into the ER using ATP, and they are directly loaded onto freshly synthesized MHC1 molecules. MHC1 is actually synthesized across the ER membrane. It's a transmembrane protein, right? So it's destined for the secretory pathway. So its peptide binding cleft is in the ER lumen, meaning we have to transport peptides from the cytosol into the ER in order to load MHC1 molecules that have been freshly made with a peptide. And they are loaded as soon as they're made, pretty much. Viruses and some bacteria grow in the cytosol, or they grow in the nuclear compartment. And in either case, peptides can be loaded onto MHC1. This is called direct presentation. This is the normal pathway, basically. Peptides that are coming from the nucleus or from the cytosol are imported into the ER for loading onto MHC1. That's the normal pathway. MHC2 molecules, on the other hand, these are loaded with a stand-in peptide in the ER, and that stand-in peptide is stripped out and replaced with a foreign peptide in the late endosome in a low pH environment. And it may also happen, recent research suggests that the loading of MHC2 perhaps also happens in early endosomes and maybe even the lysosome, and maybe even to a greater extent than we realize. So mostly MHC2 is loaded in the late endosome, but it can be loaded in the early endosome or the lysosome. Okay. Remember, MHC2 loaded with peptides taken up from outside the cell, generally speaking. Okay. And we're going to talk more about this figure later on, which is just a visual for this slide. MHC1 has only an alpha chain. That's why I say it's a one-legged molecule. And the alpha chain has 3 immunoglobulin domains referred to as alpha 1, alpha 2, and alpha 3. Not really an important detail, but maybe something to know. MHC2, it has an alpha and a beta chain. So it's 2 polypeptides that associate with one another. And each of those polypeptides has 2 immunoglobulin domains. Okay. And then, of course, there's CD1, which we'll talk about later on in this slideshow. And CD1 has only 1 polypeptide chain, and it has beta-2 microglobulin, just like MHC1. But it actually binds to lipids and presents those on the cell surface. So more on that later. So this is a figure sort of comparing and contrasting the types of locations for antigen presentation depending on the circumstance. So for example, any cell can take cytosolic antigens, load them into MHC1, and if the cytotoxic T cell recognizes that peptide, it will kill the target cell. On the other hand, macrophages can take intravesicular pathogens like certain bacteria that grow inside vesicles. It can load their antigens onto MHC2, present them to T cells that are T helper cells, and those T cells will provide interferon gamma to kill the help the macrophage kill whatever it's ingested. Okay. B cells, on the other hand, they take up antigen with their B cell receptor by receptor-mediated endocytosis. They load it into MHC2 and then they put it on the cell surface for T helper cells. Now the goal of the B cell is not to kill the pathogen, it's to turn into a plasma cell that secretes antibody that can mark the pathogen for destruction. Okay. Now there's a few interesting things that are sort of not classical for MHC proteins. On the left, the activation of naive CD8 positive T cells can occur by an interesting route in dendritic cells called cross-priming, or cross-presentation, I should say. What can happen is this. The dendritic cell can actually take up a pathogen or like a dead cell that had a bunch of viruses in it, and it can recognize that there are, well, I guess it doesn't really screen any of these proteins, it just loads them all onto MHC1. So it can take up something from the external environment, and it can actually deliver the antigens into the cytoplasm and then into the endoplasmic reticulum, where they can be loaded on MHC1. That is called cross-presentation, and that is a property that only dendritic cells are capable of. The importance of this is really large. If dendritic cells were not capable of doing this, that would mean that dendritic cells would not be able to go to the lymph node and activate naive cytotoxic T cells, CD8 positive T cells, right? So that is extremely important. The dendritic cell does not have to be infected with a pathogen in order to go and activate naive CD8 positive T cells, because it can do this cross-presentation thing. And the activation of the T cells is called cross-priming when it happens by this pathway. Okay, so this is really important to activate naive CD8 cells. Now on the right, there's another thing that can happen, but in this case, it's an interesting thing about MHC2. Normally, we think that MHC2 can only take proteins from outside the cell and then put them into MHC2, right? MHC2 is loaded only with things from outside the cell. That is not always the case. The process of autophagy, which is basically cellular eating yourself, can deliver antigens from the cytosol into MHC II proteins where they can be presented to T helper cells. So even cytosolic proteins can be delivered into MHC II, and that occurs by the process of autophagy. Now, autophagy you might have heard of, you might not have heard of it. It's basically a cell trying to consume some of the stuff that it doesn't need, okay? And it does that to conserve energy, essentially. Like, for example, if you don't work out for a long time, your muscles will start undergoing autophagy. They'll eat some of that extra protein debris that they don't need. Well, macrophages can do this as well. And when a macrophage does it, it can load cytosolic proteins into an autophagosome, which can then be targeted with MHC II molecules to be loaded and sent to the cell surface. So autophagy can allow for cytosolic peptides to be loaded onto MHC II. Okay, so both things can happen. MHC I can be loaded with proteins from inside the cell or outside the cell, although inside is more common. And MHC II can be loaded with peptides from outside the cell or inside the cell, although outside the cell is more common. Okay, so sort of weird exceptions that ought to be mentioned before we get into this. So here's the MHC I pathway in a nutshell. There's kind of 5 major steps to it. A virus infects a cell, and then viral proteins are made in the cytosol, right? That's where almost all viruses replicate. Or they replicate in the nucleus, depending on the virus, but the proteins will pretty much always be made in the cytosol. Some of those proteins are going to be imported into the endoplasmic reticulum, where they will be bound by MHC class I molecules, and then basically vesicles containing MHC I peptide complexes will be sent out from the ER to the cell surface, where they can be recognized by cytotoxic T cells, and that cytotoxic T cell will say, hey, you're infected with the virus, here's death. And it kills that target cell that had the viral peptide on its surface. Okay, so that is a very simplistic viewpoint of this, and I'm sure you're moaning and groaning, but it's going to get a lot harder. We got to look at this in a lot of detail and with our magnifying glass. So peptides have to be generated in order for MHC I to end up being loaded with peptides, right? So where do peptides come from for MHC I to be loaded with? Mainly the proteasome. You might have learned about this in cell biology. The proteasome is a multi-protein complex composed of 2 19S caps and a 20S basically proteasome. The catalytic core is the 20S component. Okay, so the proteasome generates the peptides for loading onto MHC I proteins. And it's composed of the 20S catalytic core and 2 19S regulatory caps. So the catalytic core is actually composed of beta 1, beta 2, and beta 5, and those are the proteolytic subunits. They form the catalytic chamber. They actually do the cleavage of proteins into peptides. Okay. So here's how it works. Cytosolic proteins, including defective ribosomal products, or DRIPs, are tagged via the ubiquitin proteasome system, or the UPS, for degradation. And here's how the UPS works in the cell. First, several ubiquitins are attached to a target protein that is destined for destruction. Either the protein doesn't work, or it's just not needed, or it's old, or something bad happened to it. And it also occurs at a low rate to proteins in general. Eventually, all proteins will be targeted for the UPS. And ubiquitin ligases are the enzymes that basically take ubiquitin proteins and attach them to target proteins. And they're called E1, E2, and E3 ligases. Now, the function of each of these is not super important for this class. You might have learned about E1, E2, and E3 in cell bio or something like that, but we don't care that much about it in this class. We care about immunology. Okay. Chains are going to be formed at a lysine residue, the 48th amino acid lysine, so it's called K48, on each ubiquitin protein. So ubiquitin is actually sort of like a small little protein. I think it's like 100 amino acids long. And the 48th amino acid in ubiquitin is a lysine. That's where each new ubiquitin is going to be attached into a chain. So you can see they begin to polymerize into a chain. Now, at least 4 ubiquitin proteins will be bound to the target protein in order for this to occur. Once that happens, the ubiquitin chains will be recognized by the 19S regulatory cap of the proteasome. Okay. What happens next is this 19S cap unfolds the tagged protein into a straight line. Okay. So it straightens it out like a snake that you're uncoiling, and it threads it through the proteolytic core, okay, the catalytic core. And now inside that catalytic core, the protein is shredded into short peptides, basically, and it's spit out the other end and released into the cytosol out through the other 19S cap. And now we have peptides that we can use to give to MHC1 to load MHC1 with. All right. These peptides are the source for MHC1's loading. Now, in the immune system, the UPS does some interesting things. If you stimulate a cell with interferons, each of the subunits of the catalytic core can be replaced by it's an inducible form of each of the subunits. So beta 1 and I, beta 2I, beta 5I, and that forms the immunoproteasome. This is important because the immunoproteasome generates peptides that are specifically good at binding to MHC1 molecules, because it cleaves after hydrophobic residues in the target protein, and therefore it makes peptides with C termini that are good for transport by the TAP protein that imports peptides into the ER, and also has ideal anchor residues for binding to MHC1 proteins. So the immunoproteasome is turned on by interferons like interferon gamma, interferon alpha, those kind of things. And its job is to make peptides that are super good at binding and getting to MHC1. Okay. In addition, interferon gamma, if you stimulate a cell with that, it will induce the expression of the PA28 proteasome activator complex, and that replaces the 19S caps. Okay, so the regulatory caps. This PA28 protein, it increases the peptide release rate and decreases the shredding that occurs within the proteasome. So proteins start getting flown through here at a rate of 1,000,000 miles an hour. They're just being churned over super fast without a lot of cleavage because of this PA28 protein. This means that more peptides are produced, a greater range of peptides are produced, okay? And it also can help to conserve the antigenicity of peptides that could have been lost if they were shredded down into tinier pieces, right? So if you keep the peptides a little bit longer, that gives you more opportunity to create a larger diversity of peptides so that you can load a larger number of peptides into MHC1 and get recognized by the maximum amount of cytotoxic T cells, right? So after you generate the peptides, they have to be imported from the cytoplasm into the endoplasmic reticulum. Okay, TAP1 and TAP2 are the proteins that do that. TAP stands for transporter associated with antigen processing. Okay, TAP1 and TAP2. The super duper important thing to remember about TAP1 and TAP2 is that they contain an ATP binding cassette, or an ABC. Okay, so in the cell, ABC proteins mediate the ATP-dependent transport of ions, sugars, amino acids, and peptides across membranes. So they are used for import and export of substances that normally cannot get across membranes. TAP is one example of an ABC protein. TAP, its job is to pass peptides from the cytosol into the ER so that they can be loaded on MHC1. Okay? They have an ATP binding cassette on the cytosolic side, and then they have a transmembrane domain that crosses the membrane of the ER, and then they have sort of an intraluminal side that gets rid of the peptide. So the job of the ABC cassette is to bind ATP and use the cleavage of ATP as energy to import peptides into the ER. Okay, so if you took micro, you sort of learned something similar to this with the ABCs, right? Well, this is kind of like the same thing. It's just used for transporting peptides into the ER from the cytosol. The expression of TAP proteins at the level of the gene is upregulated by interferons. And weirdly enough, the TAP genes are actually located in the MHC2 gene locus, not in MHC1, but in MHC2. So that's kind of contradictory because these are only involved in the MHC1 pathway. It's not exactly known why that is. I think in mice they're actually in MHC1 gene locus, but in humans they're in the MHC2 locus. Or maybe I'm thinking of cats or something. Now TAP, what it does is it takes peptides from 8 to 16 amino acids in lengths, and it prefers peptides that have hydrophobic or basic residues at their C terminus. Okay. Note on this, remember that immunoproteasome? Yep, that one turns down cleavage after acidic residues, meaning that it basically turns up the cleavage after hydrophobic or basic residues. So it creates substrates that are really good for import by TAP. So the immune system is kind of working together with itself in this pathway. Peptides that are too long to bind to MHC1, i.e. peptides 11 to 16 amino acids long, they can be trimmed by an enzyme called ERAP, which is upregulated by interferon gamma, but is also constitutively expressed. ERAP stands for ER aminopeptidase associated with antigen processing. So its job is to just trim down peptides that are too long to bind to MHC1. Okay. And then I also have one last note. Any peptide that does not bind to MHC1 is going to be taken back out into the cytosol and degraded. Okay. So let's look at it from the perspective of a freshly synthesized MHC1 molecule. So point of view, you are a fresh MHC1 molecule. Remember, MHC1 only has an alpha chain. So new MHC1 alpha chains enter the ER, right? They're secreted across the ER membrane because they are transmembrane proteins, where they bind to calnexin. Calnexin is a chaperone in the ER, and it binds to MHC1 to retain it in a partly folded state. An interesting note on calnexin, it is also a chaperone for MHC2, CD1, T cell receptors, immunoglobulins, and a number of other important proteins. But a whole lot of immunological proteins are going to bind to calnexin while they're being synthesized for secretion. After calnexin binds to MHC1, beta-2 microglobulin binds to a different side. It binds to sort of, between alpha-1 and alpha-3 domains. And the complex dissociates from calnexin after beta-2 microglobulin binds to MHC1. So calnexin is released. And next what happens is the MHC1 beta-2 microglobulin complex binds to a multi-protein complex called the peptide loading complex, or PLC. The peptide loading complex, it is composed of TAP1 and 2, and it's composed of 3 proteins. First, calreticulin. Calreticulin is a chaperone protein similar to calnexin, and it helps MHC1 remain folded in sort of a proper conformation, and it helps it remain receptive to peptides. Okay, so MHC1 is just holding its groove open, waiting for a peptide to swing by and slide in there. Calreticulin helps make that process most efficient. Okay. And then there's tapicin, and tapicin is extremely important, and it's probably a protein you'll want to know for sure. Tapicin is a TAP-associated protein encoded by a gene located in the MHC locus, and it forms a bridge or tube between the MHC1 protein and TAP and the membrane. Okay, so peptides that are fed through TAP and imported by TAP, they sort of shoot through tapicin and into MHC1 where they bind, and then MHC1 beds to the cell surface, right? So tapicin is the only component of the peptide loading complex that's specific to antigen processing. These other proteins also are involved in other stuff. Tapicin is specifically immunological. And then there's ERP57, also called PDI. I can't remember what this stands for. I know. Protein disulfide isomerase is another name for it. Protein disulfide isomerase. So this protein is a thiol oxidoreductase, which is a really fancy term. But really what it does is it breaks and reforms a bunch of disulfide bonds in MHC1 during peptide loading. And so it just makes sure that MHC1 forms stable disulfide bonds. Okay, and it forms a stable disulfide-linked heterodimer with tapicin, so it's directly bound to tapicin. So let's take a look at this giant complex in our first two steps that I've outlined so far. So MHC1, it is synthesized across the membrane where it first binds to calnexin, and then it binds to beta-2 microglobulin right in here. Okay, calnexin is then released, and the peptide loading complex associates. And it's composed of calreticulin, which is a chaperone. It's composed of protein disulfide isomerase, PDI, also called ERP57. That binds here, and that's going to help form stable disulfide bonds during peptide loading. And also in the PLC is tapicin. Tapicin serves as a bridge between TAP and MHC1. So, peptides that are generated by the proteasome are fed through TAP with the use of ATP, right? Where they're trimmed down to an appropriate size by the ER amino peptidase associated with antigen processing, ERAP. And now we have a suitable peptide to bind to MHC1. And it comes around and it actually moves through tapicin, although this figure doesn't imply that, but it does move directly through tapicin and into the MHC1 groove. Okay, so that's where we're at right now. And you can see this looks a lot more fruitful when it's not just a big wall of text. Okay, so that's where we're at. So we've loaded MHC1. So here's what happens next. Peptides must be loaded onto MHC1 for complete folding and secretion to occur. Without a peptide, MHC1 molecules are retained in the ER and they don't make it to the cell surface. Now, they've done experiments where they actually knock out the TAP protein from mice, or at least in cell culture, they can knock out the TAP proteins. And when you do that, you don't get any peptides imported into the ER, so MHC1 just sits there. And it actually accumulates in the endoplasmic reticulum to a really large number. And it eventually crystallizes and explodes the cell when you do that. So MHC1 is literally not allowed to leave the endoplasmic reticulum unless it is bound to a peptide. Okay, it is not allowed to leave the ER unless it is bound to a peptide. Another note: the peptide loading complex, along with the MHC1, it mediates the exchange of low affinity peptides for higher affinity ones. Okay, so there is a little bit of a competition between different peptides. The highest affinity peptides have the most tendency to be loaded onto MHC1. And that, of course, means that they have the most ideal anchor residues, and they are most in touch with some of those side chains in the peptide binding cleft, and they bind with favorable interactions with all those side chains that are facing inward. Okay, and depending on what allele variant you have of the MHC1 protein, it will influence what peptides your MHC1 proteins like to bind to. So this depends from person to person what high affinity peptides really are. The exchange and competition of low affinity peptides for higher affinity peptides is called peptide editing. And it turns out that tapicin is mostly responsible for this process. I think you can knock out all of the other genes and it still sort of works, but tapicin completely gets rid of peptide editing. If you don't have tapicin, you can't do it at all. The binding of a peptide to the partly folded MHC1 molecule releases the PLC from MHC1, so the peptide loading complex releases MHC1, and then the complex leaves the endoplasmic reticulum in a vesicle, and it is targeted to the cell surface, and it, of course, fuses with the cell membrane, and then you have your MHC1 up on the surface. Last note, any peptides that don't bind to MHC1 would accumulate if they were allowed to. So they have to be moved back to the cytosol by a protein called SEC61, which is an ATP-dependent transporter. Okay, a little bit different than TAP because this one's moving peptides into the cytosol, out of the ER. And then they're going to be just shredded to pieces in the ER, or excuse me, in the cytosol. Okay, so once we've loaded MHC1 with a peptide, MHC1 releases all the members of the peptide loading complex, and it is released, and it is secreted in a vesicle and makes it to the cell membrane where it can be recognized by cytotoxic T cells. Now you can imagine that there's a lot of targets here for viruses to try to subvert this whole thing. But remember, if a virus downregulates MHC1 on the cell surface, it gets targeted by a natural killer cell, so that's not very favorable either. So you're kind of screwed either way if you're a virus. So that's the MHC1 pathway. Haven't heard any questions yet. So I don't know. Are there any questions about MHC1? Okay. All right. Hopefully I can get through MHC2 by the end of this, and then we can talk about the weird stuff next time. So MHC2 pathway. This is an overview of that. You've seen this figure before. You're probably familiar with most of these concepts at first. So peptide MHC2 complexes, i.e. peptides bound to MHC2, they are generated in the acidified endosome, which is another way of saying endocytic vesicles, from proteins that are obtained through endocytosis, phagocytosis, or autophagy. Okay, so MHC2 can be loaded with peptides from any of these three processes. That includes, in autophagy, these peptides can be from the cytoplasm as well, right? They don't have to be taken up from outside the cell because autophagy allows this to happen. MHC2 proteins, they're first delivered into the ER membrane across pores that recognize signal particles, or signal peptides, I'm sorry. So the MHC2 protein, it is transmembrane, therefore it has to have a signal peptide at its N-terminus that is recognized by pores in the ER membrane that allow for the insertion of the peptide across, or I should say of the polypeptide across the ER membrane into the lumen. Once MHC2 is synthesized, they are transported in vesicles from the ER into intracellular vesicles containing antigens, containing peptides. For the most part, they are transported to the late endosome that has already acidified and contains only the bite-sized pieces that are the remnants of protease shredding of the antigens. But loading also occurs in the early endosome, and it also occurs in the lysosome, probably to more of an extent than we realized before, recently. So peptide source number one, this is probably the major one, is uptake from the external environment. Okay. Proteins that bind to the B cell receptor on B cells can be internalized by receptor-mediated endocytosis and then taken into the endosome. On the other hand, in phagocytosis, larger particulate materials like fragments of dead cells are taken up by phagocytes, for example, macrophages and dendritic cells, and then that can serve as a source of MHC2 peptides. Soluble proteins, such as toxins, they can be taken up by micropinocytosis, and that is something that dendritic cells tend to carry out, but macrophages also do it. And that's simply just taking up the solutes in the tissue fluid and digesting them and breaking them down. That can lead to a toxin being loaded into MHC2, or at least peptides from a toxin will be loaded into MHC2. Endosomes, they become increasingly acidic as they progress into the cell interior, and eventually endosomes do phase with the lysosomes. Both endosomes and lysosomes contain these enzymes called acid proteases, and that is basically a protein cleaving enzyme that is active under acidic pH. Okay? The endosomal pH at the end of the pathway gets down to about 3.5 to 4. And most of the acid proteases become active around 4.5 to 5.5 for the pH, okay? But they remain active down at this lower pH as well. So here's some of the endosomal enzymes. The cathepsins are the most important group. They are cysteine proteases inside endosomes, and they are acid proteases. The cathepsins are cathepsin B, D, S, and L. I don't remember what they all stand for. But cathepsin L is probably the most active and the most important. A related one is the cysteine protease asparagine endopeptidase, or AEP. And this one has been shown for the processing of only certain antigens. So if you want a T cell response to certain proteins, you do need AEP, but it's actually pretty dispensable in other cases. But you definitely want cathepsin L in general, because that does a lot of the processing for MHC2 peptides. There's also an enzyme called GILT, or the interferon gamma-induced lysosomal thiol reductase. And the job of this protein is to break and reform disulfide bonds to ensure that everything in the endosome is folded stably. Okay, no funny business while you're trying to process antigens into peptides. Peptide source number 2 is autophagy. So autophagy looks something like this. Self-antigen taken up in an autophagosome or autophagosome that is actually derived from the ER. This membrane comes from the ER. And it encapsulates some self-antigens that need to be broken down or recycled and used for energy. This is one way that cells can sort of conserve energy is by eating the contents of their own cytoplasm through autophagy. But that can fuse with MHC2 containing vesicles, and it can end up with self-antigens being loaded into MHC2. In some cases, if there is a viral infection, this can also result in viral antigens from the cytoplasm being loaded into MHC2 as well. Okay, so proteins from the cytosol like actin and ubiquitin, they are frequently bound to MHC2 molecules on the cell surface. Originally, immunologists were like, what? Why is actin and ubiquitin here? Those are intracellular. Well, later it was found that autophagy is the explanation for that. Autophagy normally delivers damaged organelles, protein aggregates, and excess cellular material to the lysosome where it can be degraded. Autophagy is constitutive in all cells, but it will be upregulated and will also take on larger structures, so it'll be more macromolecular during cell starvation and stress. Okay, stress being the key thing for immunology, right? A cell that's infected with a virus is very stressed. So autophagy will be upregulated in that under that circumstance. Okay. So some of the first steps of MHC2 pathway, they again begin in the ER, but this is not where MHC2 is loaded with peptides. Okay, it's just where it's synthesized. So MHC2 contains a signal peptide, or an SP, that designates it for translocation across the endoplasmic reticulum. Here, calnexin first binds to MHC2 freshly after synthesis, and it keeps it partly folded and retains it in the ER for a short time. That is, until a protein called CD74, also called the invariant chain, big I, little I, binds to MHC2. In this class, we mostly call this protein CD74, so that's what I'm going to refer to it as. So CD74. So CD74 binds and occupies the MHC2 cleft so that MHC2 doesn't accidentally bind to premature proteins that are being actively folded in the endoplasmic reticulum. So CD74 looks something like this. It kind of looks like, I don't know, Jabba the Hutt's tail or something, or a lizard tail. And it has this little peptide called CLIP that binds to the actual peptide binding cleft of the MHC2 molecule. So CD74, it is a transmembrane protein with the protein in the ER membrane, or excuse me, the ER lumen, and it also forms trimers using a trimerization domain. So it actually binds to an MHC2 molecule, but it also binds to two other CD74 MHC2 complexes. So you get like these big 9-chain complexes formed. So specifically, the CLIP domain of the CD74 protein is what binds to the MHC2 cleft. CLIP stands for the class 2 associated invariant chain peptide. Just know it as CLIP and just know this one as CD74. When 3 CD74 chains, 3 MHC2 alpha chains, and 3 MHC2 beta chains associate, as you see here, calnexin releases the complex. Calnexin dissociates from all these MHC2 molecules, and MHC2 is then free to bud from the ER in a vesicle. And where is it going to go? It's going to go to the late endosome for the most part. So after release from calnexin, CD74 targets the vesicle to a low pH endosomal compartment. And CD74 actually has a cytosolic sorting tag that designates its transport to this specific location, the specific endosome location. Okay, so it has a little peptide on the end of it that tells certain trafficking proteins in the cell to send it to the late endosome. Okay. After fusion with the late endosome, CD74 is cleaved into LIP22 by acid proteases. LIP22 is cleaved into LIP10. LIP10 remains bound to MHC2, and it looks like this in that case. I wouldn't worry about LIP22, but LIP10 is an intermediate step that historically students have had to know. Okay, so LIP10, so it's cleaved into LIP10 by cysteine proteases in the endosome, and LIP10 remains bound to MHC2. LIP10 actually still contains the sequence that promotes the retention in the endosome. Okay. Finally, LIP10 is cleaved by cathepsin S, or in some cells is cleaved by cathepsin L, and that leaves only the CLIP peptide inside the MHC2 molecule. And it's still associated with MHC2, so CLIP is a stand-in peptide for MHC2 during synthesis. CLIP does not contain the proteolytic retention sequence, so soon this MHC2 molecule is going to move to the surface. So how does it know to, get rid of CLIP and bind to an actual peptide? Well, the HLA-DM molecule in the endosome binds to and stabilizes empty MHC2 molecules, and it also catalyzes the release of CLIP from the cleft of MHC2. So HLA-DM binds to MHC2 and releases CLIP from the peptide binding cleft. That allows other peptides to swing in and bind to MHC2 in that empty cleft. So these peptides inside the endosome are now going to bind to the cleft of MHC2. HLA-DM also carries out peptide editing. So this occurs for MHC2 as well. Remember in MHC1 it was performed by tapicin. Well, in this case it's performed by HLA-DM. So remember, peptide editing is the exchange of low affinity peptides for higher affinity peptides. So there's a competition between peptides that can bind really strongly and those that don't bind as strongly, and they have to compete. This has the effect of lengthening the half-life of MHC2 complexes on the cell surface. Remember, if MHC2 accidentally lets go of its peptide, it's no longer going to be able to fold properly, and it's going to fall apart, and the cell's going to have to take it up and destroy it. Okay? On the cell surface, it is very likely that some peptides are captured first as longer polypeptides, and then they undergo trimming by exopeptidases on the cell surface with the active site facing outward. Exopeptidases like furin or certain other enzymes that are facing outward on the cell, and they're cleaving some of the long peptides in the MHC2 cleft. But that also further increases the number of possible peptides that can be bound by MHC2 and presented, right? So you have anywhere from 13 to 25 amino acids in these peptides, but MHC2 can probably bind to stuff that's longer than that. It just normally gets trimmed down to 25 or less, and it's going to get trimmed down to basically a random number. There's another protein called HLA-DO, and this one's present in thymic epithelial cells, B cells, and dendritic cells. And that one's a negative regulator of HLA-DM. And what it does is it binds to HLA-DM in the endoplasmic reticulum, but once it's in an acidic environment, it slowly dissociates from HLA-DM. Okay, so this is like a timed release of the protein that allows MHC2 to pick up a peptide. Okay, so you get a timed release and a timed window in which MHC2 can bind to a peptide. Interferon gamma increases the expression of HLA-DM, but it does not increase HLA-DO expression. So HLA-DM starts to outnumber HLA-DO, and therefore you get more MHC2 loading occurring in cells stimulated with interferon gamma. So they actually have higher levels of MHC2 on their surface. And that's not the only reason why. There's actually a specific compartment in the late endosome called the M2C. Not really an important concept, but they've imaged it and it's a thing. So here's the pathway in a nutshell. The invariant chain CD74 forms a complex with MHC2 inside the ER, blocks the binding of peptides and misfolded proteins. Then that buds into a vesicle where the CD74 is trimmed down to lip 10, and then it's trimmed down to clip. Then HLA-DM strips clip out of MHC2 and allows a new peptide to come in and bind to MHC2, and then MHC2 is targeted to the cell surface. Okay, so that is MHC2 in a nutshell, and I'll leave this slide for next time. So I hope everybody enjoys their spring break.